This video is going to look at enzymes and how enzymes work, and also at the factors that influence enzyme activity. So enzymes are proteins, and they're proteins that act like biological catalysts, which means that they allow reactions to occur at a lower activation energy than they would occur normally. And that's important because many reactions that happen in our body wouldn't happen naturally at 37 degrees. You need them, they would be much higher, so you need these catalysts to bring down this activation energy. And they're used to catalyze every single metabolic reaction in the body, so anything that's anabolic, so which produces new molecules, anything is catabolic, which breaks down new molecules, anything that changes different molecules from one sort to another sort, everything is catalyzed by enzymes. So let's think a little bit about enzyme structure. Enzymes are proteins, and we saw earlier that proteins are made out of a long chain of amino acids. And there's many different possible combinations, because you can have different sequences of amino acids and different lengths of proteins. And as a result, you get a different shape. And different enzymes will have very different shapes. And this shape is very specific. And the shape is specifically in such a way that it can only catalyze a specific reaction. So you don't have one enzyme doing all the reactions inside your cell. You have a specific enzyme for every single reaction that happens inside every single one of your cells. So let's look at this in a bit more of a graphic detail. So this is our enzyme, this blue Pac-Man symbol, and they have a particular shape, and part of the shape is called the active site. And the active site in this case is the cutout triangle from this circle. And the active site is where the substrate is going to bind, because the substrate also has a specific shape. So our substrate here is this kind of lime green square. And the substrate and the enzymes will collide at times. And when the substrate collides and goes into the active site, it forms the enzyme substrate complex, and the reaction that the enzyme catalyzes occurs. So in this case, this lime green cube is going to turn into a purple and a red cube, and because these two products have a different shape to the substrate, they no longer fit into the active site and they are released. The enzyme doesn't change in this reaction, so the enzyme can grab another substrate and catalyze the reaction again, and this would happen again and again and again and again. There are a number of different factors that affect enzyme activity. These are temperature, pH, the concentration of the enzyme, and the concentration of the substrate. Let's look at temperature first. So temperature, and we're going to have a colder than optimum temperature. An optimum temperature, which is the temperature that things work best at, so it would be body temperature for us generally, and then a denatured temperature. And denatured means that the shape of the enzyme has changed because the protein has at such a high temperature that the bonds that hold the protein in its particular shape have changed. So let's look at the cold one. You see the particles are moving, they're randomly moving for Brownian motion. Occasionally an enzyme will collide with a substrate, and the reaction will be catalyzed, and it's formed. At the more optimum temperature, the same thing happens, but because the particles are moving a lot quicker, because they have more kinetic energy, the product is formed more quickly. Therefore, the rate has increased. And in a denatured enzyme, they move even quicker than that, but because the active site has changed shape, you can the substrate no longer fits into the enzyme, and no reaction is catalyzed. So if we plot this as a graph, we can look at temperature as our independent variable and the rate of activity in arbitrary units as our independent variable. And as the temperature goes up, the rate of activity goes up. At 0 degrees centigrade, it will be the rate of activity is 0 because it's frozen and the water is frozen, so the particles can't move, so they won't collide into each other. As you go up in temperature, the rate of activity goes up until you reach the maximum, about 37, at which maximum rate, and then it quickly drops down and it will have completely denatured at the end. Another factor that affects the activity of An enzyme is pH. And different enzymes will work best at different pHs. So here we've got stomach pepsin, which you can see clearly there's this blue dot. And then at the bottom, with no activity yet, we've got salivary amylase and lipase that goes into the duodenum in the small intestine. We're at about pH 1.5. Stomach acid is about this particular pH. So the pepsin inside the stomach will be at its most active. As we're changing the pH, the stomach pepsin becomes less active. The salivary amylase and the duodenum lipase are starting to become more active. The salivary amylase is at its most active around pH 7. The duodenum about pH 8, 9. Anywhere beyond this, and the activity level will be very low. And this is because the enzyme denatures when it's in a pH that is not the optimum level. So by the time we get to the end, all three of these will have a fairly low activity because all three of them will be pretty much denatured. The final two factors are enzyme concentration and substrate concentration. They have an effect on the rate of enzyme activity. In this particular case, or these particular cases rather, the enzyme doesn't get denatured. It doesn't change shape, but it's more the chance of collisions that are affected. So as enzyme concentration goes up, the rate of activity goes up. And this is because there are more active sites available. And because there are more active sites available, there are more enzyme substrate complexes that can form. Because the substrates are more likely to bump into the active sites because there's more of them. Once you reach a certain concentration, adding more and even more enzyme will no longer increase the rate of activity. And that's because there's only so much substrate around, so there's going to be active sites that are empty, so at least some of the time. The final factor is substrate concentration. Now, substrate concentration goes up, the rate of activity will also go up. For a similar reason as for the enzymes, there's an increased chance of collision between the active site of an enzyme and the substrate molecule, so there will be an increased number of enzyme substrate complexes formed. And again, this is up to a certain point, at which point the active enzymes, the active sites of all of the enzymes that are around are filled with substrate all the time. If you add more substrate, the rate of activity can no longer increase, you just have more substrate waiting for the enzymes to free up again. And this is the video on enzymes, and that's all. That covers all of it.